I'm Noah Holkabor. I'm a second year at Grand Valley, and today I'm going to walk you through a little bit of the research that I've been doing in the past year. So my talk is about my use of a photographic method that is non-destructive to study the composition of a forest here in right at Grand Valley, specifically looking at their architecture, the branching patterns, the very structure of the tree. So I'm going to dive right into it. First, a little bit of background. The woody part of any tree has two main tasks. First of all, it's got to transport water up to the tree, or up to the leaves, and then it's got to support the limbs of the tree so that they don't break off. And every tree's got to do this. And some species are better at different traits than others. They optimize differently to perform them. One species might be really good at supporting its limbs, but not so good at, efficiently transporting water, and another species might be vice versa. And we can, experimentally determine this optimization of a species, or, really of an individual tree, by looking at the ratio between the mother and daughter mother-daughter ratio at a branching point. So looking here at the image, I have labeled the mother, which is the radius before the branching point, and the two daughters, which are the radii after the branching point. And we'll also look at the angle between those two daughters. Now, all the past research that's been done on this, at least what's been published, has been done destructively. So they would cut the tree down and then take physical measurements. And that's precise, but limiting. You can't go out and study an entire forest of trees like I did, because A, that'd be some hardcore deforestation, and it would just take a lot of money, time, effort. It'd be nearly impossible. You also couldn't study the same tree over time, because if you've got to cut it down to gather data, well, you can't then go get data from it 10 years later. So what I specifically did, a little bit of an overview, is first I selected a plot in the Grand Valley Ravines, and then I mapped that plot out. I took some physical measurements of trees, took pictures of those trees, then took those photos and put them into a program called ImageJ, where I could, take the measurements from the photos that I mentioned on the past slide, the mothers and the daughters, and then was just our data analysis. So first, our plot selection. As you can see in the image right there, that's my plot at Grand Valley. It's a 1/4 hectare plot in the GVSU Ravines, and a little bit about it. It's actually right near the Arboretum. It's a little bit kind of back into the woods from there, but as you can see in the image, that's kind of where it is in relation to the rest of campus. It's 25 meters by 100 meters, so kind of long and skinny, and it's partially on a slope. We didn't necessarily look at this in this analysis, but that is something we might look at in the future, is how trees at different areas of the slope are optimizing differently. And it's all deciduous trees, and I know that it's all deciduous trees because I have been to, touched, photographed every single tree in that plot. It's all deciduous trees, trust me. So now a bit more detail on the mapping process. It was measured by hand because at such a small scale and under the canopy of the forest, we couldn't really get accurate GPS measurements. And some boundary trees were marked, both with flagging tape and little tree tags, and I gathered a lot of information about them. And that's so that I could map a tree in relation to it. So every tree in the plot had its DBH measured. At least all the trees above 5 centimeters in diameter, because we didn't really count any trees below that. You'd start splitting hairs at that point of what counts as a tree. So trees above 5 centimeters. And then the location of every individual was mapped in my field notebook, which you can see in the image there. And a distance to one of those boundary trees I mentioned, it was measured with a laser distance measure, actually to 2 boundary trees, so that in the future someone could come back and, using my map and using the kind of compass, the angle and the distance to some of those known trees on the border, they could find that exact same tree again and measure it. So the meat and potatoes of this whole experiment, the photography. The first branching point, what we call node 1, is what we took a picture of on each tree. And it was photographed with a scale bar, because when we take that picture and put it into ImageJ, our image measuring software, we have to have something to, set the scale so that we know how long, X distance is. And so basically they had a pipe with a ruler on it, or a pipe that was just 1 meter long, so that later on we knew what the scale was. And you can just see some examples of some first node photographs. And those pictures are, those are actual photos that we used in our research and took measurements from. And for for some higher up nodes, because the first branching point can actually be really high up in a tree. Something new we did in this plot that we hadn't done in past experiments is we have this extending pole that can go up like 30 feet with a scale bar attached to it. So I could, go out of the forest, get it way up there, and then snap my photo with a scale bar in it. And that's helped our photos be, more, just better, higher quality. Now, the photographic method, it certainly has some challenges. First of all, there are just some limitations that are always going to be there. Taking a photo of a tree and making measurements in image J, it's always going to be less precise than cutting it down and measuring it with a caliper. We know that limitation is there. The goal is to see if we can get usable data, even though those are limitations. There's also just some difficulties with the photographic methods that we can hopefully overcome. Like I mentioned with having the extending scale bar, sometimes nodes are high up, and so we need to kind of come up with some workarounds. Or because of where a tree is at in the forest, you might need to come at it from a strange angle or be, maybe close underneath it. And some of those difficulties we can overcome, and some are just going to be there. If you look at that photo on the far right, you'll see that that's our long extending scale bar next to a 3-meter pole. So now some of the trees in the plot were difficult or impossible to photograph. That's usually because we did this in summer and the first node was obscured by branches. And so there were a couple trees in the plot where we didn't actually get the pictures of the first node because they're obscured by branches. But we do have some potential fixes for that. You can go back in winter and take those photos when the canopy is not blocking it. So there are some workarounds. And some of the first branching points are ambiguous. If you look at that photo in the lower left, it might be a little bit hard to see, but there's a dead branch here, and then we have a first node where there's 3 branches coming out at the same point, and I eventually just have to make a judgment call as to which one is node 1. And then in this other photo on the right, as you can see, there's this huge burl right there in node 1, and that might affect the data. And in past experiments or studies, I would be looking for a specific species, and I could go out and find a species that I could, or an individual of that species that I could easily photograph. Nice, obvious first node, not obscured by branches, and take a nice picture of that. But because this study was in a plot, I needed to measure every single tree. And so some of them were ambiguous or broken or diseased or weird. And so, that may have introduced, a little bit of play into our data. But spoiler alert, even with all these challenges I just mentioned, we still got workable, good data from these photographs. So let's look at a very basic level of the plot. Acer saccharum, sugar maple, and Quercus rubra, red oak, were the most dominant species in the plot by far. They both had a similar sum DBH, if you add up all the DBHs of all the individuals, but Acer saccharum had way more individuals. If you look at the charts I've got there, on the left is just the sum DBH by species, and on the right is the number of individuals per species. You can see that Acer saccharum and Quercus rubra, similar sum DBH, but Acer saccharum has way more individuals. So basically, there's a lot more small sugar maples, but there are a couple big red oaks. Okay, so looking at the whole plot on a little bit more complex level. Here we are looking at it in terms of Murray's Law, which is a mathematical model that we use to experimentally determine that optimization. And Murray's Law is pretty simple. It just predicts that at a branching point, area will be preserved. And basically that means that the area of the mother will equal the sum of those 2 daughters after the branching point. And Murray's Law is a measure of hydraulic optimization. How efficiently can that tree move water throughout its structure? So looking at this graph, on the x-axis, we have the mother values according to Murray's Law, and on the y-axis, we have the daughter values according to Murray's Law. So every point on that chart is one of the individuals in the plot. And as we can see, we've got a really strong R squared. It's pretty consistent. A slope of 1.0, and we're not that far off 1.0, would mean that a tree was optimized for Murray's Law, that it was optimized for hydraulic efficiency, and most of the trees fit on a line pretty close to that, which is what we'd expect. That's what previous literature would say that we should expect, and it just makes sense. Trees in a plot are going to be optimized to move water. But we do see a slight drop-off in the larger trees, looking up here. in that, they are not as closely fitted to the line and they're a little bit under-optimized. And that's small, but we do see that maybe they're increasing a safety margin as they increase in size. And by safety margin, I mean that when you are optimized for a trait, in this case hydraulic efficiency, the tree gains benefits, but there's also risks associated with that. So for Murray's Law, or for hydraulic optimization, that risk is embolism. So maybe it seems that when trees are a bit larger, they play it a bit safer and sacrifice a little bit of their hydraulic efficiency for a reduced risk of embolism, and embolism just being an air bubble in one of the vessel tubes that's bad for the tree. So this next chart is looking at the same situation from a different way. The previous chart, that ratio has basically been put just on the y-axis, and it's being compared against the log 10 DVH on the x-axis. And this shows, it's got a low r-squared value, that the relationship between hydraulic optimization and the tree DVH, its size is relatively independent, but there's a weak negative linear relationship. And, that's just showing, again, from a different angle, that as trees grow in size, they lose a little bit of their hydraulic efficiency, but, not that much. Now, this is a little bit more complicated and looking at the forest stand as a whole, but I'll walk you through it. So basically, area preservation, like I talked about with Murray's, a preservation from a area of higher hierarchy, the mother, to the next lowest size, the daughters. You can measure that in an individual tree, like is done in Murray's Law, or you can do it in the entire plot level, kind of treating the forest as a whole. And that's what this chart does. Basically, on the y-axis, we have on the x-axis, we have the log radius at breast height of an individual tree being compared against the log radius at breast height of the next smallest tree in the plot. So each one of those points is a species, an individual being kind of compared to the next smallest individual, as a way of comparing how the size distribution changes throughout the composition of the plot. And we see only a very small divergence. It's pretty tight on that line. And that's just indicating that area is being preserved in the entire plot. And we see a little bit of drop-off again with the larger trees being a little bit farther apart and a little bit off of the line, at least more so than the smaller trees. And that kind of anecdotally makes sense. If you walk through a forest, there's going to be a lot of small trees that are really close to each other in size. But if you think about it, the biggest trees in that forest, they're probably going to have a bigger difference from each other than maybe the smallest trees would. Now this is taking that graph that I just showed you and putting it up on the left, same graph. And then on the right, we have a comparison of individual trees in the plot, very similar to Murray's Law, but even simpler. It's just looking at the log mother area on the x-axis versus the log daughters area on the y-axis. And those charts look very similar. And that's because they have some similar components, even though the one on the left is looking at the entire plot and treating it as one whole, and the chart on the right is looking at each individual tree and treating it as an individual. But both of them are based off of radius measurements. The one on the left being measurements I physically took at breast height, and the one on the right, the log mother area, being measurements taken from node photographs. But it's similar measurements there, radii, based off of diameters, and it's looking at a similar principle of this area preservation at a point of going from a bigger thing to a smaller thing, a different level of the hierarchy. So these are actually analogous to each other. They are measuring similar properties, which is wild and kind of hard to wrap your head around it, but it's indicating that our forest stand is behaving as a hierarchical branching network, thinking of it similarly to a tree. It's mimicking kind of this optimization that an individual tree does, and that sounds wild, but it makes sense, and the forest stand is really trying to maximize the amount of light it can capture, right? The trees are competing to get light. But an individual tree is also trying to maximize the amount of light that it can receive. And so at a certain level, those similar goals are leading to an optimization in how the mass is distributed. That's not necessarily because of biology, and is more because of physics. And so I'll bring it a little bit more concrete again and talk about some species-specific analysis from the plot. This graph right here is a comparison of the uniform stress model, which is another mathematical model that we can put the ratio of mothers to daughters into, but it is actually looking at the optimization for the structural support of the tree, or of the limbs, and it's compared against Murray's ratio, or Murray's law, either way, which I've already talked about, hydraulic efficiency. And it's looking at the top 5 photographed species in our plot. And if, say on this chart, each one of those colored lines is an individual species, and all the individual points are individuals in the plot. So if a species were to have a slope of exactly 1 on this graph, it would mean that it was equally optimized for Murray's ratio and the uniform stress model. It was equally optimized for hydraulic efficiency and for structural support. But we don't see any species that do that. In fact, almost all of them are more optimized for Murray's law, for the uniform stress model, whereas only Quercus rubra was more optimized for Murray's law. So most of the species, at least these 4 that we're looking at, the 4 of them were more optimized for structural support, and only red oak is more optimized for hydraulic efficiency. And to kind of clear up some clutter, I'll look at just Acer saccharum and Quercus rubra, the 2 dominant species in the plot. This is that same graph, just with some of the things removed. And we see that they optimize very differently. They are the dominant species in the plot, but Acer saccharum is really more optimizing for the support of its limbs. It's more towards the uniform stress model, and Quercus rubra is optimizing more for hydraulic efficiency. It's tilted down towards Murray's ratio. And for each of the, for those optimizations, there are some benefits to the tree. It's better at transporting water, or it's, better at supporting more massive limbs. But there's also costs and safety margins. Maybe we see them optimizing differently because a tree can't be perfectly optimized for both. Maybe to be optimized for hydraulic efficiency, you've got to sacrifice a little bit of your optimization for the support of limbs. And there's also safety margins to consider that I briefly mentioned earlier. Taking the uniform stress model, if you are perfectly optimized for that, you're going to have some big limbs, but they might be more likely to break off. And looking at the Murray's law and hydraulic efficiency, if you're perfectly optimized for that, you're going to be moving water super efficiently, but by kind of being close to the edge, you're at an increased risk for embolism. So, optimization is good for that trait, but there are risks associated with it, and there are reasons to not be perfectly optimized for something. And the relationship that I just showed you between those species in the model, that's not a size thing. Looking at this chart here, we have Acer saccharum and Quercus rubra. They take up a similar some deviation in the plot, but the average Acer saccharum, the average sugar maple, is way smaller. So maybe it's just that sugar maples are small, and so that they optimize this way. Maybe it's just small trees optimize this way. Well, no, it's not. We ran that regression there on the right, each of those points being one of those 5 species that I mentioned, and look at that, there's not a significant relationship there in any shape or form. And that's comparing that ratio that I mentioned earlier to the log 10 DBH, basically comparing the ratio between Murray's law and the USM and the size of the tree, and there's no relationship. So it really seems that there is a species difference there, not just a size difference. These optimization differences are due to them being different species, not just due to a different size composition. And so the overall conclusions of everything I've talked about, first and foremost, most important thing, photography can produce valid data. There are limitations, it's messy, it's not perfect. Cutting down a tree would give us way more precise data, but we can still get valid, useful data from photography, and that's a new thing for this field. Second, all the species in aggregate, they're pretty close to hydraulic efficiency, And that's exactly what we'd expect. And that just lends more credence to photography as a valid methodology. Third, the forest stand as a whole is optimized to fill space in a way that is similar to how an individual tree fills space. And that's not due to, that's not necessarily due to straight biological factors. That is because they're trying to perform similar tasks. They're trying to capture as much sunlight as possible. And so due to physical properties of the universe, they optimize similarly, even though one is looking at an individual tree at a branching point, and one is looking at the distribution of mass in an entire forest. I think that is kind of wild and fascinating, and I'm still trying to wrap my head around it. And finally, looking at the species level, we see that Acer sacrum, sugar maple, and Quercus rubra, red oak, the two dominant species in the plot, are optimizing in very different ways. Sugar maple is more looking for structural support of the limbs, and Quercus rubra is more optimizing for hydraulic efficiency. And those differences, as I just talked about, they're due to size differences, they're due to species differences, not size differences. And so, before I finish up, I just want to, cite the papers that I've been looking at, and also thank Gary Greer of the biology department for excellent mentorship through this entire process. Not just for this Student Scholars Day presentation, but I've been working with him for over a year now, and I'm going to continue to work with him in the future. He's been an excellent mentor, and I've loved doing research with him. And I would like to thank Grand Valley State University, not just for the opportunity to present today at Student Scholars Day, but also for the funding in that my summer work, when I went and took all the photographs of these trees in the plot, that was funded by Grand Valley. And without that funding, there's no way that I could have been able to get all that data or invest as much time and energy into this project. So again, thanks to Grand Valley. And finally, thank you so much to you for watching and listening to this project. If you have any questions or comments, I would love to hear them. There might be a way to do that through the system set up for Student Scholars Day. Otherwise, my email is up on screen right now. I would love to hear from you. Or if you're watching this video on YouTube, feel free to put them in the comments down below. Well, thank you so much for listening to my talk. Again, feel free to reach out and go look at some of the other Student Scholars Day projects. There's a lot of good stuff out there. But thank you very much. I hope that you enjoyed the rest of your time at Student Scholars Day.